Hello, everyone, and welcome to today's webinar titled Highly Efficient Therapeutic Antibody Development Solutions. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Antonina Salcido of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please go to genscript.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located to the right of the abstract. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located within the navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Amanda Grimm, Senior Segment Marketing Manager at GenScript USA, Inc. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Amanda, welcome. You may now begin your presentation.
Thank you so much. Hello, everyone, and welcome. As mentioned, my name is Amanda Grimm, and I am the Senior Segment Marketing Manager for Antibody Therapeutics here at GenScript. My presentation today is titled Highly Efficient Therapeutic Antibody Development Solutions. In this presentation, I will provide you with an introduction to GenScript, discuss what are antibody drugs, outline the development workflow highlighting GenScript's end-to-end solutions at each stage, and then I'll wrap up with a summary. Might have to edit that out. For those of you not already familiar with GenScript, we are a leading global synthetic biology company with offices around the world, including Oxford, the Netherlands, Singapore, China, and Japan. We were founded in 2002 in Piscataway, New Jersey, here in the USA, offering the first custom gene synthesis platform. Last year, we celebrated our 20-year anniversary, commemorating our growth as the GenScript Group, adding subsidiaries like Legend Biotech that specialize in cell therapy, Custom Array, which is our Seattle-based oligo and production array facility, and our CDMO, GenScript ProBio. As we've evolved, we've also added our catalog products division where you can find automated instruments and ready-to-use reagents. So let's get started and talk about what are antibody drugs. Antibody drugs are based on the magic of our own immune system, whereby we produce and engineer antibodies to treat or modify disease. On the left, you can see the types of antibodies our immune system naturally develops. The antibody drugs are highly specific to their target, the most common type being a monoclonal antibody. As development proceeds, monoclonals are modified in order to improve function, and this can include a number of different format modifications. We can see some examples down here on the right. One of these is an antigen binding fragment, or Fab. Their small format makes them great for either therapeutic or diagnostic use. One popular Fab therapeutic is used for preventing blood clotting. We're starting to see more complex formats like the bispecific T-cell engager, referred to as a BiTE. These are popular formats in cancer therapeutics and are a bispecific monoclonal antibody that have two different targets, where one is for the CD3 receptor on T cells and the other is tumor specific. Another type of complex format is the trifunctional antibody. This targets 3 epitopes, and we're also seeing these emerge in the cancer therapy development industry as well. Looking back at the history of antibody therapy development, the first FDA-approved antibody was approved in 1986. Since then, there was a slow progression between then and 2006, whereby after 2006, the FDA started approving new antibody drugs on an annual basis. In 2021, the FDA approved its 100th therapeutic antibody, and I'm sure we're all familiar with this one as it was the monoclonal antibody therapeutic for SARS-CoV-2 infection. Here we have a table of the top 10 FDA-approved monoclonal antibodies. The leading drugs target cancer and are seen highlighted in red. These include treatments for melanoma, colorectal cancer, and non-Hodgkin lymphoma. Not far behind cancer, we have therapies for autoimmune diseases. These are shown in purple. Treatments listed in this table include rheumatoid arthritis, psoriasis, and Crohn's disease. A smaller percentage of therapies cover other issues such as bone loss or infectious disease. I want to share one newly approved antibody that came out in November 2022. I'm personally really excited about this one since type 1 diabetes runs in my family. It is an autoimmune disease that attacks the insulin-producing cells in the pancreas, where the disease outcome is that the individual eventually becomes dependent on insulin injections. This mAb treatment targets the CD3 receptor on T cells in order to reduce the effector function of the immune attack by those autoreactive T cells. When it's administered to early diagnosed patients, the treatment has shown to be able to delay disease onset for as long as 25 months. So let's take a look at the workflow and what goes into the development of antibody drugs. Antibody drug discovery is the process of identifying and developing antibodies as therapeutic agents in the treatment of various diseases. The process involves the identification of target antigens, selection of appropriate antibody formats, optimization of antibody-antigen interactions, and rigorous evaluation of safety and efficacy through the preclinical and clinical trials. The goal of antibody drug discovery is to develop safe and effective drugs that can selectively target and neutralize those disease-causing agents while minimizing off-target effects. Here, I've created an illustration of the overview of the development broken down into these 4 stages. And in the next slide, we'll discuss some of the listed development issues that researchers may encounter. So I've highlighted some of these as barriers where GenScript can help provide a solution for notably achieving sufficient quantities of the target protein and achieving sufficient selectivity and specificity of antibody drugs. In these next few slides, we'll go through the workflow stages independently, and then we'll discuss solutions that will help make your discovery and development more efficient. So starting with target identification, the goals in this stage include understanding the disease mechanism, selecting the therapeutic target, and then validating the target. I've listed out here the different areas, gene analysis, protein analysis, and cell-based assays that GenScript can provide solutions for your project. I won't be covering all of these, but we'll highlight a few of our most popular solutions. A reminder as we get into the discussion, if you see something you're interested in and I don't cover it, please don't hesitate to reach out. I made sure to include my contact info at the end, and we can also take questions afterwards. So starting with gene analysis, many of our customers utilize the Gene Titan oligopool service because it allows them to screen a large number of genes at the same time. The oligopools are mixtures of oligonucleotides, which can be designed to target specific genes or gene families. Researchers can knock down the expression of multiple genes in parallel, which can help to identify which genes are involved in a particular biological process or the disease state. They can also be used to perform genome-wide screening, allowing for the identification of novel targets or pathways that may have been previously considered. Using oligopools can be more efficient and cost-effective than screening individual gene knockdowns, especially when a large number of genes need to be tested. Some researchers run into issues expressing their target proteins. Our protein expression service will help ensure that your protein is expressed in sufficient amounts, whether you use it for antigen, for antibody generation, or for downstream screening. These proteins can be produced using a variety of expression systems, including bacterial, insect, or mammalian cells. For in-house analysis, we also offer gels and blotting automation as fundamental tools for protein detection and analysis. This includes the blotting units, membrane processing instruments, electrophoresis standards, and high-quality antibodies for screening. From a personal standpoint, anytime you can automate Western work is a must. I've spent a number of hours and time in my career making gels, dealing with uneven transfer, and millions of trays and tubes just for staining. As part of the target validation process, cellular analysis is commonly used to clarify the target role in a signaling pathway or its ability to bind ligands. Stable cell lines are a valuable tool in antibody drug target validation that provide a consistent and reliable source of cells that express protein at a known level. This enables researchers to evaluate the function and identify potential drug candidates that selectively target the protein. Our cell line solutions include a collection of ready-to-use stable cell lines, or we have the capability to develop a customized line specific to your project. We offer hundreds of GPCR options, most of the immune checkpoints for cancer therapy development, various SARS-CoV-2 proteins, and the Claudin lines as well. So once we've identified and validated our target, we now move on to generating and optimizing our leads. In this stage, our goal is to generate a number of hits, identify those that bind our target through screening, and then optimize our most promising candidates. This slide shows the comprehensive list of GenScript solutions in this area. So let's look at a few of our key products and services. Mouse hybridomas are the traditional method of generating monoclonal antibody hits. They're still the most common approved therapeutic. Our MonoExpress service provides multiple options to generate high-quality leads in as little as 13 weeks. We've made this very simple, and all you have to do is provide us with your target sequence. We'll take care of the protein expression, we'll prepare it, and we'll immunize the mice. Our MonoBoost immunization is an optimized technology that will expedite your hybridoma project compared to the standard method, generally eliminating about 3 to 4 weeks of development time. We offer 2 additional methods of antibody lead generation using B cell screening and cloning that improves the timeliness that you achieve usable hits. Our MonoRab monoclonal antibody service uses the rabbit immune system to generate and isolate B cells post-immunization. Rabbits are a known and often preferred animal for their high affinity and avidity antibodies. The service line shown on the top is a simple enrichment of antigen-positive B cells from isolated PBMCs that is then followed by screening and harvest. The high-quality leads are then sequenced and engineered for recombinant antibody production. To further expedite your timeline, we also offer the Beacon system in combination with rabbit B cell generation. The Beacon allows for high throughput screening since it can test a high number of B cells and perform multiple screens in parallel. Screening that typically takes a week or more can be completed in a day. The high quality leads from this testing is their isolated sequence and then engineered for recombinant antibody production. For our customers who have completed development to this point and want to continue to do so on their own, we do offer do-it-yourself options for purification systems that will allow you to automate these processes. Our AMAG assay provides high-throughput antibody or protein purification using the magnetic bead technology. It can purify up to 12 samples, and purification is complete in 1 hour. Our newly launched AMAG Quattro provides a high-throughput automated plasmid purification solution. The instrument has a modular capability, allowing you to add up to 4 instruments that can be controlled from one device. The 4 modules can handle up to 6 samples each, totaling 24 samples that results in yields that range from 300 micrograms to 1.5 milligrams of purified plasmid. Once you've identified your antibody lead candidates, optimizing will provide several benefits in terms of improving the potency, selectivity, and pharmacokinetic properties of the drug, which will ultimately increase its efficacy and safety in patients. GenScript offers several optimizing capabilities, including antibody affinity maturation, FC engineering, stable cell line development, humanization, and more. Some of our further optimization tools include our precision mutant libraries. The libraries are a powerful tool that enable the identification of high affinity antibodies with desired functional properties. The libraries are created using site-directed mutagenesis to introduce a defined set of mutations into the target protein or antigen, which can then be used to screen for antibodies with improved binding, affinity, specificity, and functional activity. Precision mutant libraries can be generated for various targets, including the difficult to express proteins and membrane proteins, and can be used in combination with our phage display technologies for antibody screening and selection. GenScript's TurboCHO platform is a proprietary technology for antibody drug development that utilizes a high-performance CHO cell line to improve protein expression and yield. We have 3 different levels of service that are applicable to the different stages of antibody drug discovery. The platform offers several advantages over traditional CHO lines. This includes higher protein expression levels, improved cell growth, viability, and reduced production costs. In addition to the cell line development, the TurboCHO platform also includes a range of services for antibody lead optimization. That includes gene optimization, vector construction, and antibody engineering to improve the affinity, specificity, and other properties. Having identified your leads with high potential, preclinical studies are then conducted with the goals to perform trials on non-human subjects and evaluate efficacy, toxicity, and PK. My slide here shows the solutions available at GenScript, including capabilities to file and support you through the IND studies. The products outlined in this slide include our endotoxin detection kits. These are used to detect the presence of various endotoxins that are usually cellular or bacterial toxins. These can cause inflammation and other adverse effects in patients. The kits are important to ensure the safety and quality of preclinical research materials, such as recombinant proteins and antibodies. We additionally have an endotoxin removal kit available as well. That one is a column-based kit that can be used to remove the endotoxins prior to use in animal studies. GenScript also offers a variety of immune checkpoint proteins and antibodies, which are essential for studying the immune system and developing immunotherapies. Immune checkpoints are regulatory molecules that help prevent overactivation of the immune system, but they can also be exploited by tumors in order to evade the immune surveillance. By targeting these checkpoints, researchers can potentially enhance the immune response against cancer and other diseases. Our immune checkpoint products include recombinant proteins, high-quality antibodies, and ELISA kits for detecting checkpoint protein expression. Our ProBio team provides comprehensive services to support preclinical studies for antibody drug development, including services related to the chemistry, manufacturing, and controls, or CMC. They offer a range of these services, including cell line development, process development, analytical method development, and manufacturing of clinical trial materials. Their manufacturing capabilities include microbial fermentation, mammalian cell culture, and protein purification. They use state-of-the-art equipment and technologies to ensure high-quality, consistent production of your antibody and other biological molecules. In addition to CMC and manufacturing services, the ProBio team also offers a range of analytical and characterization services to support the preclinical studies. These include assays for protein purity, identity, and potency, as well as stability testing and pharmacokinetic analysis. The final stage is moving your new antibody drug onto clinical development. The goal of this stage is to conduct clinical trials in people according to phases outlined by the local regulatory body. In the US, the FDA outlines 3 clinical phases, where phase 1 is a small group of patients and normal volunteers. Data is collected to assess dosing, side effects, drug metabolism, and pharmacogenic actions. After passing phase 1, phase 2 includes a larger group of subjects and studying the effectiveness for the indication, identifying any short-term side effects, and also any risks. Passing phase 2 moves to phase 3, where an even larger subject pool is studied for effectiveness and safety, evaluating the risk-benefit, as well as determining the basis of physician labeling. The clinical phase is complex and requires strict regulatory adherence. Our clinical development support is provided by our highly experienced CDMO team at GenScript ProBio. They offer expertise in cell bank generation, scale-up, tech transfer, and regulatory compliance to help accelerate the development of your antibody drugs and ensure their success in the marketplace. The cell bank generation services include developing master and working cell banks, ensuring regulatory compliance, and performing extensive quality control tests to ensure the consistency and stability of the cell lines. In addition to the cell bank generation and scale-up, ProBio provides technical transfer services to ensure seamless transfer of manufacturing from the research lab to a clinical manufacturing facility. There's no wait time to get started, and they're able to complete the technical transfer within 2 months. They work closely with the clients to ensure that all technical and regulatory requirements are being met. That brings us almost to the end. I've covered quite a lot, so let me run through the next couple slides in order to summarize. So let's review the workflow. Ultimately, we want to create an antibody drug that's going to be safe and effective. It's common to encounter barriers along the way, for example, screening a large group of potential target genes, protein purification and analysis, and making sure you achieve high-quality antibody leads. To solve these, in particular, we looked at GenScript's oligo pools, the automated instrumentation, and our B cell sorting and cloning options, just as some examples. Here's a summary chart of our antibody drug-related services. Some were covered in my presentation today and some were not. You'll see highlighted in the chart the quality production levels and capabilities for each service. We are quite flexible, and at any point we can easily adapt most of these as needs change with your product or with your project. So this concludes my presentation. I want to thank you again for your attendance. I've included a QR code to our antibody drug discovery webpage, and feel free to reach out to me if you have any questions about what I went through today or would like more information in general. Thank you.
Thank you, Amanda, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Now let's get started. Our first question here is, can you provide more information about the custom cell line development?
Yes, of course. So for the custom development, we prefer customers provide their own cell lines. The service itself includes DNA construction, transfection, selection of high-producing clones, and then we'll follow that up with testing for the protein expression, stability, and functionality.
Great, thank you. Another question here is, what are the detection levels of the endotoxin kits?
Great question. So we do have both qualitative, yes, no, and also quantitative versions. The quantitative kit, yes, quantitative. The quantitative kit plots standards. So with that, you're going to generate a standard curve. And once you've generated that, the kit can then detect a range between 0.01 and 1 EUs per mil.
Thank you. Our next question, how is the monoboost immunization different from a traditional immunization?
Yes, so the monoboost technology is proprietary technology developed here at GenScript. What I can say more about that is that a typical timeline is that antibody using the monoboost is going to be ready in about 8 weeks with this option.
Time for a few more questions here. Our next one is, can I send my already expressed and purified protein for antibody development?
Yes, definitely. So we can generate antibodies with your protein, but just keep in mind it might change some of the project timelines, but definitely.
Thank you. We'll wrap with this one. How many samples can the AmMag do in a day?
So with a normal workday, you could get a minimum of 60 samples, and that's based on putting in 12 samples and the fact that it can complete it in about an hour.
Awesome, thank you. And thank you, Amanda. Do you have any final comments for our audience?
Just thank you so much. And as mentioned, we're here to make your research easy. So please reach out with any questions that you may have.
Great. Thank you again, Amanda, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.